Okay. First of all, I'd like to thank each and every one of you guys for showing up, and each and every one of you guys for investing and taking and being a part of this venture with us. I'd like to let you know that I have read all of your bios and all of your comments. So I recognize each and every one of your names, and just want to let you know our level of appreciation for you taking part of this venture with us. Okay, so like I said, my name is Dr. Maria Nagy, and I'm the CEO of Quorum X Diagnostics, and we have developed the first fully at-home COVID antigen test. Results not in 15 minutes, but in 9 minutes. We are now going to be the 9-minute people. It's kind of like a pregnancy test, but for COVID. Our mission is simple. We want to get a COVID test into every single person's hand that wants one. We have yet to do that. I don't have to tell you that we're in the middle of the worst pandemic of our lives. There's 25 million, almost 26 million cases worldwide, and 850,000 deaths. And in the US, there's 6.2 million cases and 188,000 deaths so far. When we're looking at the solution that we have going now, there's 3 different types of tests that you see in the market. These tests are the antibody test, the PCR test, and the antigen test. The antibody test will measure to see if your body has produced either IgG or IgM antibodies. This is your body's response to an infection. So that development of those antibodies can take days to weeks after an infection has started. So it's very good at telling you if you've ever been exposed, or maybe at the very tail end of an infection, they might capture and say, oh, look, your body is responding to that infection. This involves a finger prick, and the cost of that can be anywhere from $60 to $200. And depending on the company, the test accuracy can be from 60% on up. The second test you hear a lot about is the PCR test. This is what you see when you're going through that drive-through. So what they're doing with that test, they're not looking for antibodies, but they're looking for a piece of RNA. So you look inside the virus, they'll pull out a subsection of the RNA and amplify it to where you have about 1 billion copies. And that can take about 5 to 6 hours in the laboratory. You have to have very expensive equipment, a very highly trained staff, and results can take between 1 to 2 weeks because there's such a backlog of people waiting to get tested. These tests cost between $140 and $200. And then let me tell you why this 1 to 2 week waiting period is such a problem. Let's talk about Jane. Jane works in a coffee shop, and somebody got exposed at her job. And so the boss says, Jane and everyone else, she sends all the employees home. So Jane is now sitting at home. She needs to be working because otherwise she can't pay her bills. She calls to schedule a test. When I called to schedule a test, it took me 2 to 3 days to get something scheduled. Then when I went to the PCR drive-thru, I actually sat there for 3 hours. Some people sit and wait for up to 7 hours. Results can take anywhere from 7 to 14 days to get results. So here's poor Jane. She unexpectedly had to stop work because of this COVID scare. She realizes she has no food in her fridge. Now, even though she knows she's supposed to be quarantining, she has to sneak off to the store to go get some eggs. So 14 days later, when she gets her results, they're negative. So she's like, okay, great, I can go back to work. But in actuality, since she snuck off to the store, her results are invalid. So technically, she needs to go back and get another test. This is the problem with a 1 to 2 week wait for results. They're almost null and void. Let's talk about our test. Our test is a simple antigen test. So instead of looking for antibodies or looking for an RNA, we're looking for protein on the exterior of the virus. Without this protein, this virus cannot attack the body. So if the virus is there, the protein is there, and we can scan for it and tell you immediately if you have an infection. This is an at-home test. There's no specialized equipment. You need no prescription. You need no appointment. You can either go to your drugstore or get one mailed to you. And then you can run the test on yourself and get a result in 9 minutes. We have an at-home, we have an app that's complimentary you can download, and this app will allow you to take the test, which looks like a pregnancy test. And once you get results in 9 minutes, you're going to be able to take a picture, and our app will have AI technology to analyze the line to ensure that you're not getting a false negative. So let me tell you how it works. You receive the kit in the mail. You follow the directions and swab your nose. There's a video on the app that will guide you to make sure that you're swabbing your nose carefully. And it's only a nasal swab, not a nasal pharyngeal. So about 1 inch in your nose, not 3 inches. You put it in the tube, you agitate it, you drip it on the stick, and then you take a picture. Not only will you get results in a few minutes, but it will allow you to connect directly to a telemedicine service. For this, we built an app, and here are our first mock-ups to the app. You'll see that there's an embedded video. There's steps laid out. And then once you take the picture, your information will number one get shared anonymously to the appropriate local and federal agencies. We'll be able to make a heat map, a real-time heat map, in terms of where the infections are developing, what parts of town. This can all be done anonymously, just based on your zip code. The results, you'll have the option of sending a certificate to your employer or a wedding event or your friends and family as you see fit. And then you can immediately click a button and connect with a telemedicine service. So that's what we are building. Let me tell you about market share, because we're all investors here, right? So the total, if we look at the number of Americans, according to the 2020 census, there's 300,000 Americans in the 300 million Americans according to the 2020 census. If we look at the number of people that are between the ages of 20 and 90 years old, that's 220 million. If we focus in on the number of people that are in the densely populated urban areas, that's 183 million. If in there, 3% of those people decided to buy our test and only purchased one test at an average price of $37.50, our total potential revenue is 205 million. Another way of looking at this is that if we sell 2.7 million kits at $37.50, we will make 100 million in revenue. Companies with 100 million in revenue typically have a $1 billion valuation. So if we can achieve 2.7 million in sales, we can put ourselves in the category of a $1 billion valuation for a company. But there's competitors. Everyone's concerned about the competitors. The Quidel, the BDs, Saliva Direct, Abbott, Chorus. So let me tell you about what they have and what they're doing and why we're different. Okay, let's start with the Yale Saliva Direct test. This is a PCR test. It's an at-home collection kit. So they send you a box, you spit in a tube, you package it, and you mail it to the laboratory. It's still a CLIA certified laboratory. There's a bunch of equipment. It takes several days to process, and then they'll eventually mail you back the results. In terms of Quidel, BD, Abbott, and Chorus Bioconcept, these are our antigen-based tests. These were just recently, they just released these antigen-based tests. So they do look like these lateral flow tests. The results are in 15 minutes. But with Quidel, BD, Abbott, Chorus, and Sorrento, they are all urgent care, doctor office, they're point of care tests. So you still need healthcare professionals to help you with the test, and you need a CLIA certified laboratory to run the test. With our test, the QXD COVID-NOW test, it's fully at home. You need no equipment. You need a cell phone, and you get results in 9 minutes. There's no PPE, there's no lines, there's no, you don't need a prescription. You just do the test in the comfort of your home, and anonymously you can upload your information and report it to the agencies that need to track the development of the virus. We've got 3 go-to-market strategies: B2C, B2B, with the drugstores, and then to industries. So B2C, that's direct to consumer. You're going to be able to order this kit from our website or through Amazon. And then we have B2B through the drugstores. We're working with Physician 360. They are a telemedicine service located in 50 states, and they have access to 900 plus drugstores with drugstore kiosks already in place. So Physician 360 is already selling their flu test, their strep test, and now they can add their COVID-NOW or our COVID-NOW test right next to it. Lastly, we're going to pull together a sales force that will focus on meatpacking facilities, government agencies, schools, daycares, bars, and restaurants. Our wholesale price will be $25, and our MSRP retail price will be $50. Our production and shipping costs are presently around $5 to $6, so our gross margins in any of these scenarios are pretty good. Let me tell you about our traction. So we've been working on this product since April, and so in the last 4 months, we've built working technology, we've built the app, we filed a patent, we've applied for NIH funding, we've filled out an IRB and applied. We've done some branding and logo, brand new branding logo website. We've discussed a deal with Physician 360. We have a letter of intent for 20,000 units. And the next 2 major steps are clinicals and EUA. And we've done all this for under $100,000. Just imagine if we had a lot of money, how fast we could go. Let me tell you who's gotten it done, how we've been able to achieve this. This team is awesome. Let's start with Dr. Angel Rivera. He's the one that's helped develop the technology, wrote the patent, the NIH, the IRB, anything science related, he's the guy that's gotten it done. Dr. Rodney Nash, he's one of our advisors. He's helped us secure the lab space, gotten us patient samples, helped us with the PCR testing. Mr. Azad Rahman, he's our all business guy. He knows nothing about science, but we need one of those guys. He's helped us with the marketing, the website, the branding, and the logo. Next, we have Mr. Eric Woods. He's a PhD candidate and a former patent examiner. And he's helped us with the plastics manufacturing, negotiation, and also the patent writing. Lastly, we have Dr. Navi Jita, and she's a MD, NIH postdoc from Emory, and she's helped us with the technology and the writing of all of our documents and reviewing anything that's written. So this team has been amazing. They've all stepped up and just been amazing. So let's talk about funding. We have now raised a total of $150,000 between private and WeFunder funding. We are looking to raise a total of $500,000 so that we can finish up our manufacturing, our clinicals, and our FDA EUA application, and a couple of other bullet points as listed below. We're raising this money on a $3 million valuation on a safe. And then lastly, another thing to discuss is, what does the future look like? There's a number of things we could do. There's an opportunity, as with most companies, that after the development of our COVID-NOW product, we may appear, we may be a good candidate to get acquired by another company. If we look at Sorrento Therapeutics, when they announced the development of their antigen test, their market cap doubled from 2.3 billion to 4.4 billion in 1 week. Another route that we can go is that we can fully develop our technologies, because not only do we have the COVID-NOW technology, we have a second technology that I can tell you about later. It's the fastest for pseudomonal pneumonia. If we build out all our technologies and get 5 successful products, and we have designs for all of them, then we can be similar to Alair. Alair was acquired by Abbott back in 2016 for $5.3 billion. And then, of course, there's the opportunity for an IPO. So these are the various situations that we are keeping our eyes open for, and we hope to develop our technologies out so that we can be similar to Alair. That's our goal. So that's what we have. That's what I have right now for today. And before we finish, I'd like to bring on Dr. Angel Rivera, and he's going to give us the scientific data for our technology that we've collected so far. So if you just give us a minute, we will change places and bring up his presentation. Thank you. I see some of you guys already typing in some questions, Paul and Gary. So we're going to address these questions as soon as Dr. Rivera has finished presenting. So feel free, anybody that's watching, to go ahead and type your questions in, and we will stay until the end until we've answered every single one of your questions. So with that said, I'm going to go ahead and set up Dr. Rivera's presentation. And that should just take just a minute. Okay.
Good evening. I hope you guys can hear me. I'm Dr. Angel Rivera, and I will be presenting some of the preliminary data that we have about COVID-19, COVID-NOW, testing COVID-19, and let me fix this for a second, guys. Can you see that?
Give us just a few minutes.
I can see that. Can you go to the first one?
I swear it worked perfectly the 3 times we practiced this earlier today. Okay.
So there we go. Hopefully you guys can see what I'm doing. We wanted to show you guys, let me see here. Okay, so the first slide is QXD-NOW. It's a lateral flow that will detect antigen that comes from the virus. This is in a home kit. It will detect COVID-19 antigen, and especially for asymptomatic patients, but it can do asymptomatic and symptomatic. These patients are very contagious at this moment, and we want to detect them very quickly, not a week after. So this is very fast. Basically, we email it to you, you receive it, you swab your nose, put it in PBS or saline or whatever we are sending to you. You shake a few times, put it in the bottom of the lateral flow, and then turn your phone on with the app that we have developed. You wait 9 minutes, take a picture, and then you can say it's positive or negative. And any questions, you can send it to Physicians 360, or you can just call one of us and we can tell you. So basically, the lateral flow to the left is a real patient, is positive, so we'll have only one line, and the one in the right is a negative, so 2 lines. It's very clear. It shouldn't be any problem to understand, and this is within 9 minutes. So what we did was we obtained 18 frozen samples for patients that have been confirmed by PCR to be positive. Here we tested each one of them with our lateral flow, our QXD-NOW, and 18 out of 18 were positive, giving us a sensitivity of 100%. Now we wanted to answer the question, will it cross-react with other bacteria that you might have that produces pneumonia, or negative is negative? And basically, we try many of the bacteria you can see in this table. We also have more here. And at the end, out of 21 samples that we tried, 20 were correct. One of them was positive, and we think it was false positive because of the pH of the sample was incorrect. Therefore, specificity at this moment is 95%. Now we wanted to know a time course. What is the optimal time to determine if you are positive or negative for COVID-19? So we started put the 4 drops at the bottom of the lateral flow and start timing at 3 minutes. So 0, 3 a picture, 6 a picture, 9 minutes a picture, 12 minutes a picture, and so forth and so on until 1 hour. And we saw that between 9 and 12 minutes, these are the ideal times, 9 minutes up to 12 minutes. It gives you exactly negative when it's negative and positive when it's positive. So this created our 9-minute test. So basically, we can conclude that 18 out of 18 were positive, hence our sensitivity is 100%. The negative 20 out of 21, 95% specificity, no cross-reactivity with any of the bacteria that we have untested. 9 to 12 minutes is the optimal time. It's very easy to use. 9 minutes is simple. Smartphone app, which AI technology that can tell you if you are positive or negative, and eventually will give you a copy number, so how much virus you really have. And this is obviously an essential diagnostic tool to help combat COVID-19. Thank you so much. I hope you guys enjoy it. Here's Dr. Nagy.
Thank you for Dr. Rivera for stepping in and telling us the science. And that is the presentation that we have for today. And I will now open it for Q&A. I already saw a couple of questions in the queue. So I will get prepared, and then we will stay for as long as you guys have questions. So please feel free to continue adding more and more questions. I see quite a few of them in here. And give me just 1 second. So the first question is from Paul. Are you able to manufacture at a large scale, or is it possible at this time? So we have talked to the plastics manufacturer as well as the lateral flow manufacturer. They can start at 40,000 a week and ramp up to 1 million tests a month within a month. And then they will be able to continue producing that level, and they have overflow capacities as well. So they are quite prepared to produce at a large scale. The next question is from Gary. He said, how is the antigen different from the antibodies versus contracting the exposure versus contracting the virus? So when you're looking for antibodies, you're looking for antibodies that your body has produced in response to the infection. For instance, let's say you have the flu. When you get exposed on day 1, by day 3, you're going to have symptoms. And then by day 5, day 7, day 12, your body will start producing antibodies so you don't get infected a second time. Or if you do get infected a second time, your response is much lower. So these antibodies are called IgG and IgM, and they get produced as your infection is going away. So when you hear the antibody test, that's what they're looking for. They're scanning, has your body produced IgG or IgM? So with an antigen test, we're looking at the virus. An antigen is just a fancy way of saying protein, or it's a fancy way of saying protein. So on the exterior of the virus, there's a bunch of proteins. And so we're scanning to look for one of those proteins. So we're looking for the actual virus when we're scanning with our lateral flow test. And hopefully that answers your question. So, and it's a real-time assessment of if you're infected or not, as opposed to did you get infected earlier earlier in the week, in the month, or earlier in the year. Okay, the next question that I see are, what are the qualifications needed for test subjects for the clinical trial? So we just got, if you want to join me, we just got finished writing our IRB. And so we just finished writing our IRB, and the IRB is the form that you fill out in order to be able to do clinical trials. You have to include a number of inclusion and exclusion criteria. And so our inclusion criteria are the people that we're allowed to be test subjects are you have to be between the ages of 20 and 90. Go ahead
Right.
and speak
You
to
have
it.
to be an adult. We don't want any children at the moment. They have certain criteria that we wrote to for the qualifications. But once the IRB has been approved, which it should be a few days from today, then we can start the clinical trials immediately. And we have manufactured 300 lateral flows to start this immediately.
Yes. Another inclusion criteria or exclusion criteria is we will look at people that have experience using diabetes test kits and things like this. We'll let them try the test, but we have to monitor how many of those people. We want to focus on people that don't normally use these
Tests.
these types of tests.
We want to see how simple it is for people that have never done this.
Yes, we're going to have to analyze the instruction manual, the app simplicity, the ability to do the test. So we're measuring a lot. We're measuring usability as well as the ability of the test to measure the infection. So, oh, wow. Okay. Have you ever collaborated with DARP for this testing security, especially with even National Guard pandemic? Okay, so we have never collaborated with DARP. That is something we could look into in the future to see if we can acquire some of these military type contracts. But if you have some contacts and you want to give us an introduction, we'd be open to that.
That would be great. Thank you.
Okay, it says, are you going to retest? This is from Edward. Are you going to retest, recertify the sample which may have had the pH off in order to improve specificity from 95 to 100? Well, we tried to do that. And so what it is once the pH is off, then you can't, what we would have to do is tweak the pH. And then we don't know what kind of chemistry occurred when the pH was thrown off. So we won't be able to retest that sample.
It's only one particular patient, but we will have 300 other patients that we can increase the specificity to the sensitivity much higher. I mean, sensitivity is 100%, but the specificity, we can increase it to almost 100%. It's just one particular patient that's something wrong when they collected the sample.
Right, and at the time, we didn't feel comfortable throwing that data out. So, okay, the next question is from Paul, and it says, does this test need any FDA approvals? Yes, I mentioned EUA, but I guess not everyone recognizes that acronym. So right now, every test on the market, there's probably about 140 or so tests that are on the market. 140 of them are the PCR kind of tests, and then a handful of 25 are antibody tests, and then I think 4 of them are antigen tests. So whatever that total number is. So anyway, those are all under emergency authorization use approval from the FDA. So typically with these kind of medical devices, it's a class 2 medical device requiring a 510K, but under a pandemic and this emergency situation, you need an emergency authorization. So instead of them taking 3 plus months to review the data, they review it in about 2 weeks.
Yeah, 1 or 2 weeks we have an answer back. So once we have the clinical finish, we will submit the data, and a week or 2 after that, we should have FDA approval.
Right. So EUA approval, yeah. FDA EUA approval. They also make it much easier because they provide a template and they lay out exactly what bullet points that you have to hit and what data numbers they want. So everything's laid out to make it much easier for the developers to achieve those goals. Okay, the next question is from Chris, and he says, is there a way to reduce IgG and IgM that causes inflammation like steroid and prednisone.
Yeah.
Perhaps.
So usually they're treating the patients now with steroids, as you mentioned. It's a little more complex than just IgG, IgM. We have what is called a cytokine storming, where the patients have so much protein called cytokines that they drown in fluid, because the purpose of cytokines is to invade the area and try to kill the bacteria, but it's killing you too. So yes, steroids is one of the protocols that we're using today, and it does work.
Okay.
Okay, good.
Okay. The next question is from Dr. Lee. You may have mentioned this earlier, but is there an intellectual property associated with Quorum COVID-NOW test? Yes, we have. We have filed, we are patent pending. And then we foresee applying for international PCT utility patent as well. But that'll be just a couple of steps down the road. Next question is from James. He says, have you considered developing an affiliate program, webmasters and bloggers to market the test for a percentage of the sales and price? Wow, that's a very interesting idea. We have been talking with a production company, a movie production company, and they did offer to promote our product if and if they're able to use it in their filming process or production process. So we have had that conversation. However, they finished their filming before we had a chance to get the product to them, as we're doing clinicals now. But that's a very interesting idea. We were hoping to do some kind of grassroots kind of marketing on this product. So I hope that answers your question, but we will keep that in mind. Let's see. And from Joe, we have, how would your product be involved with the COVID vaccine dosages to be available around November 1st in recent news?
I guess I would go on to challenge the patient once he has a vaccine with some artificial, not so powerful virus. I'm not sure exactly the protocol yet. And then we could use our lateral flow and look at it, the COVID-NOW, and look at if the patient has an indication of the antigen. However, most likely the vaccine will have the antigen there too. So it could be used to see if the vaccine is working, and it could be used to see the virus is there, but it's attenuated, so it wouldn't hurt it. So that's a good thought, and yes, the answer is we could do that.
Okay. Okay, and we have another question here by Dr. Lee. When is this new raise going to happen? Well, we're presently, in terms of fundraising, we're going to continue raising on WeFunder until the 15th. I believe that's the day, September 15th. It is closing. So we're actively in raise right now. This is one of the venues that we're raising, and I'm actively pitching to other outside and private investors as well. So if you would like to invest more, you like to talk to me privately, you're welcome. I think my contact information is on the website, or you can just go ahead and invest through WeFunder and continue investing that way. And then we have another question right here from Malika. Okay, it says, will the test be able to tell if a person is asymptomatic versus a person with some symptoms?
Very interesting. So I guess it will depend on how much virus you have. So a symptomatic patient could be a patient that has a low copy number or low amount of virus. The immune system hasn't seen the virus yet, but our test is so sensitive that should be able to see it, and the AI, should be able to tell you the copy number. A symptomatic patient usually are very, very sick. They have a lot of virus, so we should be able to see the difference. In theory, we haven't tried, but the answer is yes, we should be able to see a difference. On the other hand, you can have a asymptomatic patient that is super sick and doesn't know it, but that's rare. It does happen though. But then to answer your question, yes, we should be able to tell copy number and therefore symptoms versus no symptoms.
Okay, to expound on that a little bit, when you have asymptomatic people versus symptomatic, the symptomatics are going to be the people that are coughing. They're going to be the ones that have the fever. They're going to be the ones that are not feeling well in various levels. It could be a little cough. It could be you're in the hospital. So that's the range of symptomatic patients. And then in terms of asymptomatic, they're carriers, they're viral carriers, and they are spreaders. But they don't have the cough, they don't have the fever, they don't have the headache. They're just walking around feeling like they're normal, but they are actively spreading. So in situations like the story I told you about Jane is an asymptomatic person. She has no symptoms, but because somebody at her workplace showed up positive, Jane had to go get a test, even though she's asymptomatic. And so our test would be appropriate for asymptomatic patients as well, because they're carrying a viral load that's high enough to get somebody else infected.
Right. And for us to detect.
And for us to detect. So, okay. The next question is from Joe, and it says, if someone wants to get involved as a sales rep for your product, you can give me an email and we can go from there.
There we go. Thank you.
Thank you.
Yeah.
Thank you.
Give us your info.
Yes. Okay. And the next question is from Chris. And the question is, what if you know how many persons have been infected a second time? Will the test be effective? What if you know how many persons have been infected a second time? Will it be effective?
If you're infected the second time, will make no difference. We can see it. We will see it because we are looking at the presence of the virus and the protein that the virus is making, or proteins. So you're infected 3 different times, 4 different times. As long as the virus is there, we will see it.
Right. And it's also dependent on how many copy numbers are you carrying around so yeah, and now there are new reports or some reports coming out that some people there are cases of second infections. Yes. So we'll see how that develops over time. And then we've got another question right here, and it says, will you have this presentation available at WeFunder to view? Absolutely. We're going to take the recorded presentation and upload it to WeFunder. And we may actually have a second presentation for those that weren't able to attend. And that may happen in another 2 weeks. Just so once we decide on that, we'll give you updates on WeFunder as well.
And feel free to contact us for any questions.
Oh, yeah. And you're welcome to contact us at any time. Okay. And then from Gary, it says, how will you solicit the test subjects for clinicals?
So we are contacting our friends that we know, and they know friends of friends of friends. So through right now talking to friends, we probably do Facebook, we do WeFunder, we do other alternatives. But right now we're talking to people that are talking to people. We are almost to 100, maybe more people at the moment.
Right. To expound on that, so for clinicals, you are allowed to post on Facebook, and we put into our IRB that we're going to use Facebook, WeFunder, and signs, and you can post. We will be having an active waiting list coming soon. And so essentially what the clinicals will entail is everything will happen anonymously. You will have about 30 plus or so questions, a questionnaire that you're going to do about various components about your symptoms or related to COVID. number one, and then number two, questions about the usability of the test and such. You will do two parts. You will do a swab. You will take a picture. Some people will actually have to record themselves taking the test, just to make sure that they're able to swab their own noses properly. It's been shown that people can swab their own noses and that's acceptable, but we want to make sure that you guys are able to use our product and swab your nose properly. And then the second part of it is a number of questions about usability, about the app, about the test, about the tube, about the instruction guide. And then everything will be uploaded into the cloud anonymously. And then so the first swab will be where you're running the test yourself, and that picture will be taken in real time. We will have a copy of that. And then secondly, you'll do a second swab and mail it to us, and that will be tested for PCR, right? And the science, yeah.
So everything will be PCR verified.
Everything will be PCR verified. Okay. And then the next question is from Paul. Would you recommend testing them more than one time?
Sure.
I'm not sure if you're referring to clinicals or if you're referring to people testing more than one time. We foresee, or we suspect, people will be testing more than one time, because I know in the last few months of this virus, if there would have been a test on the market, or I could have gone down to CVS, I know I personally would have tested at least 3 or 4 times very easily. So, and last one is from Tom. He said, and I want to share this with you guys. He said, great job. Just a comment to all participants. I invested $50,000 into QXD. This will save lives and make the world a better place. And Maria and her team are second to none with their passion to bring this to market. This is an excellent opportunity for you to make a difference in the world, a potentially significant return as well. If any investor is on the fence with their investment, I'd be happy to talk to you. Feel free to send me, Tom, an email, and he has his email. And I will try to share it with everyone in the chat. It's dolezelt87@gmail.com. Wow. Thanks. That's very kind of you. You're awesome. I'm going to try to send it out to everyone in chat. I believe that this is, oh, it is in chat.
Yes.
No, it's not in chat.
Yeah.
Here we go. And I can also.
Thank you, Tom.
Thank you, Tom. We appreciate that. So with that said, I want to take just 1 second and give you a quick peek at our brand new website, although we are tweaking it, but hopefully you can see this and just give you a very quick tweak at what our website is looking like. There are obviously some things we need to, some spelling mistakes that we still need to catch, but look at this state-of-the-art world-class website that we will be launching with soon. So, and of course, I have not grown a beard, so that is not my picture. And then one last thing that I did not mention, but I'll mention right here. We do have some more technologies. Hold on 1 second, I will show you. And I won't go into a lot of detail, but right now we have the COVID-NOW test. We have the QXD-1 fast test for pseudomonal pneumonia, and this test right here is also ready to go to manufacturing, to do clinicals, and we've done an FDA pre-submission and an international PCT utility patent on this. However, we halted the development on this so we could focus on COVID. And then lastly, we have designs for a whole myriad of other tests as well. So you're not, it's not, COVID Now is not our only product. We've got several other products as well in the pipeline. So with that said, I'm going to see if there are any other questions. Do you guys have anything else? Any other questions? Oh, wait, I see two more. Home testing. Oh, referring back, I think Paul is referring back to his question about should you test more than once. We believe that, you know, it should give you an accurate result. And then if you're concerned later on down the line, you get a second exposure, then you could go do a second test. So.
Yeah, if you feel like you need to, sure, you order it and it will be delivered to you.
Yes. And then another very nice comment from Dr. Lee. It says, great presentation. Thanks for your time and effort. So thank you. Thank you for attending. So and then lastly, can your testing for the presence of the S spike protein also eventually lead to therapeutic medicine that has a reaction to the spike, rendering it useless as a fusing edge for COVID-19 infection? Right now, we're just focusing on diagnostics, diving into the development of a therapeutic. That's a whole nother world. So right now, we're going to stick to what we know, which is diagnostics. And then who knows what the future holds. But that's our plan for now. So we have now, it's now 8 o'clock.
Any more questions?
Are there any other questions? So I think what I'm going to do now is thank every single one of you guys for showing up. Thank you for your investment thus far. I hope you decide to invest more so that we can continue and finish this out and run faster and hit the finish line sooner.
Yes.
And we're enthusiastic to have you guys as partners with us. If you have any more questions, feel free. I've sent, I think, everyone a thank you email. And so you have my contact information. Feel free to reach out to me or Dr. Rivera, and we'll be glad to answer any other questions that you have.
Yeah, thank you so much, guys. Oh, still another question?
Stephen, thank you. And, oh, one more question. I'll be glad to stay. We'll be glad to stay as long as you want. Everyone else is welcome to leave. But if you have more questions, we will stay. So this question is the business model eventually add models to the testing base? Is the business model eventually add models to the testing base? So we have one model. We are in the development of our first model that you're going to see is the COVID Now co-branded with Physicians 360. That is our telemedicine service. So you will see the test with the telemedicine component. Later on, we're going to do a second test that, without the telemedicine component, because I'm sure some people don't feel like they need the telemedicine component. So we're going to have those two models as our starting models. And then we'll go from there. So, okay. Yes. Okay. Oh, wow. A whole bunch more questions. I'm staying here. We're staying here. There is a crowdfunder testing various insert models. I'm sorry, for whatever reason, Chris, your question is not coming through very clearly. So if you can retype it, I'll be glad to answer it. Okay. Johans. Oh, wow. More questions. Hey, Well,
It's a recording of this.
will there be a recording? Yes. Yes, absolutely. We're going to post it to WeFunder and you're welcome to watch it. And I think we're also going to do a second presentation in about 2 weeks. So, and then lastly, the accuracy of the test. No worries. We're excited to say our preliminary results.
Sensitivity is 100%, specificity is 95%, and it will get better.
So that's our preliminary results based on 39 patient samples. And then so now we're going into clinicals, and there we're going to look at somewhere between 150 and 300 patients, depending according to the FDA requirements. So, okay, another question. I may have missed this earlier. Can you tell me if I can get the test now? Not yet. We need to finish EUA. We need to get EUA approval before we can officially disseminate the test. Right? And thank you.
Thank you.
Chris. And then Stephen. Oh, to try it out. You're welcome to sign up for our clinicals.
Yes.
Those will be anonymous. And so we will be posting details about that on WeFunder and a couple of other different outlets coming soon. Okay. Okay. We hear you, Chris. We hear you. Okay. Is there anything else anyone wants to see again, or anything I can repeat? Just want to say thank you to Bob, Brian, Chris, Daniel, Gabrielle, Greg, James, Joe, Mark. Hi, Mark. Paul, Sherry, Stephen, Tal. Hi, Tal. Tom, and Johan that are still on. Thank you so much. When there's another question? Oh, yay. Mark, thank you very much.
Thank you.
Thank you, Mark. We are excited of our pivot as well. It was needed. It was the right thing to do. And luckily, we were very lucky. We got lots of preparation, and the opportunity presented itself. And so we were able to take our knowledge and our relationships and apply it to COVID. So thanks a lot, Daniel. We appreciate it.
Thank you, Daniel.
We appreciate you as well. Thank you.
Thank you.
And Paul, thank you.
Thank you.
All right.
There we go.
Hi, Mark. Yeah, pneumonia is a huge space, and we are ready with that product as well. We just put it on hold. We just put it on hold because COVID just, took over the world. So, thank you. One more. From an investor point of view, handling the test might encourage greater financial involvement. I would like to try it in pre-clinicals if available to do so. Yeah, Stephen, just reach out and we'll go from there.
Yes.
Thank you. Wow. We appreciate it. Any other questions? Let's see. Oh, yes. Okay. $3 billion market cap in 12 months. Yeah,
Sure.
sure.
Why not?
Need a couple more team members and we'll get it done. Absolutely. We're ready for the race. Thanks, Stephen. Appreciate it.
Thank you.
Appreciate it. And I appreciate every single one of y'all's comments. I've read them all. I've read y'all's bios. So I appreciate each and every one of you guys. So
Thank you,
thanks, Sherry. Appreciate it.
We love you too.
Thanks for all your help. So let's see, any more questions? Comments? We'll stay till the end. Sherry says, way to go, Angel.
Oh. So thank you guys.
Yeah. Hold on. We'll just stay. Thank you, Tal. So thanks, Joe. Thank you.
Thank you so much.